So moving a little bit away from the lab and more to the ebb and flow of Michigan's winter waterways through the perspective of P. clarkii and its survival. This is the lazy one, right? I'm not going to need it.
No, there's another one.
That's all right. We're first going to go over a little bit of the biology, the background of P. clarkii and its abiotic tolerances. The winter simulation and its protocol, we'll get into that in a minute. The statistical and graphical analysis of that, and also just the future research and where we're at currently. I'll try not to spoil too much. Let's just jump right in. So, Procambarus clarkii is a cambric crayfish, and it has a whole lot of names. There's the red swamp crawfish or crayfish, both are acceptable. Louisiana crawfish or crayfish, and my personal favorite, mudbug, which sounds like it was named by someone's uncle. These things aren't native to Michigan in general. They're native to more of a tropical regions. They're not supposed to be here. They're from the Gulf states. Think Gulf Coast, Florida Panhandle, Louisiana. Not an accident in the name there. However, they've been introduced, and that is where we get the term alien versus invasive. If a human has put something somewhere, that is going to be alien versus just them walking itself, which is invasive. It's been all over the world in Asia, Europe, and Africa, and part of Louisiana's culture is crayfish. If you remember, there's the Cajun culture and stuff like that. And because of how well these grow, they've been introduced almost all over the world. These are found in almost every tropical global area in the world. In Africa specifically, they're used as a biological deterrent. So schistosomes are a type of parasitic flatworm, and they affect humans, and if you've seen the people with kind of bloated stomachs, that's usually from a flatworm infection. The flatworm, the schistosome, uses snails as one of their vectors for its life cycle. So if you introduce P. clarkii, which eats just about everything, into it, it will fancy a snail and help decrease the amount of parasites that are in the water. Besides just being tasty, and I've actually tried them, they are kind of delicious. If you've ever been anywhere where they serve crayfish, the big red ones are probably P. clarkii. They're extremely tolerant invertebrates. These things have not only survived basically all over the world, but they've thrived all over the world. In areas of dryness, they're a burrowing species, and so they can go into that during desiccation and survive up to 4 months basically without water, just kind of crammed in their little tunnel. They can also survive pretty severe hypoxia, less than 1 gram per liter of dissolved oxygen, which is death for most, even down to 0.75, which is basically death to anything but P. clarkii. And because of where they are from, there's a lot of estuaries and general saltwater, freshwater, meaning they have a slight tolerance to salinity. 35 parts per trillion in general, which doesn't seem like much, but if someone adds salt to your water at a restaurant, you'd know immediately. Like I mentioned earlier, they are a burrowing species, and so they tend to find somewhere a waterway, and they will dig tunnels around it, which is excellent for the P. clarkii survival. Not excellent if you're trying to craft a very careful irrigation system. So they love to run amok, basically in and out, destroying those on purpose, obviously, but in general, they'll just kind of dig wherever they can. This has also sometimes caused erosion to the level of a population destroying dams and actually eroding them away and causing the area to flood and disrupting all of kind of the biological organisms that are living there. Their constant digging also kicks up a lot of soil, which we kind of refer to as a nutrient loading of the water. You add a lot of nutrients in there. This then causes cyanobacteria to grow there in large amounts. We call those blooms. And if you have too much of that, you have eutrophication, which is essentially too much algae where there's really not supposed to be that much algae. Besides digging, it's also not super nice to the crayfish around it. Maybe not on purpose always, but it is known as a vector for Aphanomyces astasi, which is kind of known by us weird crayfish people as the crayfish plague. This is a fungal infection that has killed, honestly, almost all of Europe's crayfish. They have no immunity to it whatsoever. P. clarkii isn't affected by the infection. However, the infection can be spread by it. It won't kill it off, but it'll spread it to its crayfish friends. In general, besides spreading disease to its friends, it also tends to accidentally outcompete them. These things are very, very voracious hunters, and just in general, they eat kind of anything around them, plant matter, animal matter. They've been known to hunt amphibians and even small aquatic birds. These things are vicious, and as someone who has to handle them, usually with hands, yes, they're very vicious. For that reason, when they're growing up as baby P. clarkii, they will kind of eat whatever available, and they will just keep eating and keep growing almost uncontrollably, which causes them to then outcompete other species around there, especially in areas of warm weather, kind of our tropical regions. This happens to bump into Orconectes propinquus, which is going to be the control for this project, the northern Clearwater crayfish. And I think this best illustrates the size difference. This is actual size, comparatively, not in real life, that'd be terrifying. But you see little, I gave them all names, so most of them are dead now, but in general, I gave them all names for the time. And we have little Valeria here, which is somewhere around 2 or 3 centimeters, maybe even smaller. And that was actually our largest Propinquus. And on the right, we have the adorable Pixie, which weighed in is somewhere around 76 grams, and all of its fighting force. So this is kind of the general, if you saw one of these in the water, we're talking our little things versus these. So you can see how they maybe outperform them. Like I said, their habitat is tropical. They're not supposed to be here. They will move kind of around. They've actually been known to move a kilometer or two in search of more water. But in general, they don't travel too much farther north. The most northern area, until very recently, that they've been found thriving is in southern Ohio. So kind of as we approach Michigan, not terribly close, but we're getting there. And so, and this is one of my favorite things I've read about this. There was a blogger a couple months back, when they were found recently, compared them to the xenomorph of aquatic species. If you were familiar with the 1970 movie Alien, that's the terrifying creature in the top right. And obviously this is ridiculous. But I really liked all of his parallels in that they will show up in an environment, realize, okay, this is nice, adapt to it, and kind of take over, which is something we see in this species really anywhere we put it. So it's very dangerous for anything around it, and like I said, the irrigation systems. However, we know most of how the P. clarkii live from its molting patterns in the food industry, actually, since it is made pretty much commercially around the world. We look at its molting patterns. How quickly is it growing? We need to commit more of this to maybe a big bundle of crayfish for this meal. And we notice you don't want to grow them in cold. They tend to grow very, very slowly. In fact, sometimes not enough energy with the molting actually is too strenuous and it kills them. And according to the Atlas of Crayfish in Europe, which is a real thing, about 2.5 degrees Celsius is kind of the killing temperature of the species, and that's at least what it says in the Atlas. So it makes you wonder, this kind of invincible creature, with its danger to biodiversity and irrigation systems, infrastructure worldwide, what can stop it? What thing could prevent this from invading an area, and you think possibly cold, just judging by what we know of it commercially. Now, native species in general experience problems with the cold. I would say most of you don't enjoy the cold, just in general. And it's the same with any other species, especially the native ones, where you notice more fatality while they are living in the winter. But we don't know whether or not this species can survive all. And I hate this stock photo, but I think it best represents kind of what my project would do as a whole. So the question is would this Procambrus clarkii be able to survive somewhere north of its normal habitat, say a Michigan winter? And so we had to make a Michigan winter. We're going to make more of it, unfortunately. But within parameters. At the time, we had no running water, and I mean that by we couldn't have moving water to simulate a river. It was just kind of in a box. So we had to compensate for that by making the water slightly above freezing. We did check to see if you just froze the whole thing solid and put the crayfish in it. It died, so we couldn't do that. But we looked at weather temperatures kind of within the last couple years. When it was started, I looked at about 5 years. I figured that was probably a good sample for it. Looking at our longest winter, we defined winter by about 4.9 degrees, which is what we could do in the lab without parts of it freezing, because you don't want it to be a slush sickle either. We had looked at about 140 days for our longer winters, 50 days, which I don't believe for a second in Michigan, as our shortest winter within that 5-year span. And then the average winter, which was somewhere in the ballpark of 2 or 3 months, or around 100 days, with a slight variation on temperature somewhere around 4.9. So we had to make these conditions, and this is literally what the process looked like. It's a bit light in here, but what you see is several bins in a refrigerator, and I assume we were supposed to originally have food in the lab or whatever materials we needed. I decided to put living things in it. And I allocated the isolated P. clarkii and propinquus, which we used as our control, which we know this thing survives here, so obviously it has something going for it. In about 2.8 of dechlorinated water, the chlorine is very dangerous to the crayfish. We don't want to put that in there. That kills most things in general. And kept it oxygenated, which was actually very, very helpful because it would keep the water kind of moving and prevent this kind of slush effect where the water would move from higher and low density, getting colder or warmer, especially since it was sitting in a refrigerator, the surfaces tend to be cold. We kept this at about a constant temperature, somewhere resembling that of just a Michigan winter at 4.92, a little bit of variation there. And also an automatic light cycling. You wouldn't think this would be super important, but activity in crayfish is very much influenced by night and day cycles. In the lab, we have red lights and daylight and stuff like that, representing when these things will be active and not active. And also to simulate famine, I fed them every 3 to 5 days, erring on 5. In the lab, we usually feed them every 2 or so. So I wanted to kind of simulate, well, this would be a winter condition where they're probably not going to find a lot of food, so let's put it towards the end of starving versus the end of luxury. So, looking at our preliminary results. And I say preliminary because this project, and pun completely intended, moved at a bit of a glacial pace. It took quite a bit longer for the species to not survive than expected in P. clarkii, and so our sample sizes were quite small overall, as it took like almost a year to get what little amounts we had. So these are very much preliminary, and there's a lot I would love to do in the future, but we'll look at what we have so far. This is my favorite graph, because it proves me the most right. So what you're looking at here is the overall days survival on the control, O. propinquus, and then P. clarkii, which is the experimental in this situation. And we're looking at somewhere around 40 days. I'm going to try this again. Cool. Okay. Somewhere around 40 days with P. clarkii, and somewhere over close to 120. These are on average and compared with an independent t-test, finding them statistically accurate despite the small sample size. We also looked a little bit just at P. clarkii. We wanted to get some information while we were freezing them to death. We looked a lot at the mass and the length of the species as we're going them. Like I said, it's not a huge amount, but we've noticed kind of the average size was kind of the average survival. That's looking also at as they're growing. We want to look at maybe infants might die immediately when put in the cold, and maybe these older ones are old and molted many times, and possibly not surviving very well as well. So we look at how the mass affects longevity and length, looking at mostly just these smaller ones, kind of average-sized ones, showed the most trend. So looking at the actual cold trials, we see that the positives of the whole thing is that your average winter, given our information earlier, is going to be more than the max survival of any of the P. clarkii we tested, which is great. This is definitely proving the hypothesis in that situation. And also, nice to notice, the shortest winter, which I still don't believe, is still longer than the average P. clarkii, suggesting at this point they can't survive on average, even in our shortest winter, which I think that was just seriously an anomaly. I don't believe 50 days. Looking also at how they were living, checking vitals, stuff like that, we noticed a significant decrease in aggression by the P. clarkii. Like I said, most of this handling is done by hand, as opposed to netting or something like that. You don't get them tangled and overstressed, so you kind of very quickly move them places, and they're not a big fan of that. So they will sometimes draw blood. So a significantly decreased aggression, and also significantly decreased defecation as well, regulating activity. Looking at the negative, we had a really small sample size. We had a control of like 4, and we had a experimental of like 12. But there was not much we can do about that, judging by how slowly they died. And it was also a question of whether or not that represents the population. Also looking at water temperature, our lab has the tendency to pull in cold water as opposed to warm. Who knows, they might be more adjusted to cold by that point, and that could be an issue there. My personal favorite, the curious case of Vincenzo. Yes, I named all of these. And he is that big red bar we see up there of 92 days that doesn't represent really the sample at all. Most of them lived around a month and a half to 2 at the most. This one lived almost 3. That could be a huge issue considering that is actually longer than the shortest winter. So future issues, I'd love to have more samples. 30 would be great. That's kind of the statistical, you should use this much. And also, more time and more room for it. There's still a lot of information that can be gathered by just watching them freeze, I guess. We need a bigger refrigerator, more time, or essentially a bigger box. This is a burrowing species, so possibly burrowing is a way of insulating themselves from the environment. There's been no data that suggested that in any of the papers that's been read, but in general, they might protect it. So I'd love to do some kind of real-world trial tunnels. And what makes them tick? During the freezing, for lack of a better way of putting it, we want to look at their metabolic analysis. And this is possible to get all of our information. They use a lot of isoenzymes, which are enzymes that function at a specific temperature well, and then they also have kind of a flip that functions at a lower temperature well. We also have our lactate dehydrogenase, which we know is one of those isoenzymes which deals with glycolysis during the time. Looking at climate change, which you saw this coming, I'm sure. This is a real thing. There is climate change, so no need to have a cow over that. But we see it's also accelerating, and we increased the temperature of 0.2 between 1990 and 2010 by 2%. That's in Fahrenheit, I know, but trust me on the conversion there. We also see species that are not supposed to be going places. For example, the genus Anopheles, which is the malaria-carrying mosquito for the Plasmodium. And if these things are moving to these areas, why not P. clarkii? Well, possibly P. clarkii. As of last summer, they actually found 2 isolated populations in southeastern Michigan of unknown origin, so we're not sure if these have just been put there or they're possibly someone dumped them. They're huge in the pet trade. And we also have seen clones of the species, parthenogenesis, which you're unfamiliar with, is kind of the female producing eggs that are unfertilized but still viable young. And there have been clones of P. clarkii found similar to the marbled crayfish, which is essentially taking over Europe and Africa right now. So with this additional adaptation, you wonder, how can these be stopped? And how are they going to be provided in the scientific community? And I would just like to say that in general, I think Jeff Goldblum put it best, that life finds a way. I'd like to acknowledge Dr. Dan Bergen, my mentor, and Samantha Mackey, who is the assistant, and also the Crayfish Neuroscience Lab. Let me freeze things for a little while. The Cell and Molecular Biology Department, as long as the Biomedical, because I actually did biomedical research, and the DNR, because they are actually the only ones letting me have this. Our lab is currently the one licensed to hold and experiment on clarkii. So, since they're so invasive. And also, thank all of you for being here today.
So I know that you said that you saw them obviously dying as they were freezing and stuff. And that you
I didn't watch.
Right, no. You analyzed that data and stuff. But you said that you took some vitals and everything. Did you see any cool results in brain activities or heart rates or anything?
We did not do heart rates.
Okay.
Overall, it was pretty much things that are cold move slower. So
Right.
I didn't include it for that reason.
Yeah.
But we kind of went more so on the metabolic process, how much waste they're producing. That was a better indicator. To even see it, one box was totally clear for the clarkii, and you looked at control and you couldn't see into it. This thing was moving all over the place. So.
Yeah. Thank you.
Yeah.
So where did you get your clarkii from, since they're not native?
So they are shipped into us. And I'll be honest with you, I'm not sure. I'm guessing it's from a wide range of things. Like I said, they're in the food industry, but you're not allowed to experiment on them because they're afraid of them being released into nature. So we actually have kind of a under the table deal with the DNR about they have to bring, if they bring them in, we're the only lab licensed to actually study them for the risk of them getting into the environments. What's up?
How many did you have in each box? And
Isolated. It's just one.
okay.
Yeah.
And then was the fridge closed? How did they?
Yeah. So I had to take the picture with the fridge open, or else it would be a terrible picture. But yeah, for the most part, it was closed. There was kind of a big note on it that said, "Please don't open this. Science is happening," or something to that effect. So people weren't supposed to open. I'm sure there was variations. People had to really quick run in and grab, because we feed them tilapia, which is held in the in the refrigerator, so they grab it and stuff like that. But hopefully not too much temperature difference from that. What's up?
So the living
You know it's you. There's just all things.
so the living conditions of your test were just in water?
They were just in water.
Okay, so you said that they're known to burrow for up to 4 months with little water. Have you thought about putting sediment into your experimental design?
So that was what I was talking about kind of with the future experimentation. We didn't really have a way of doing that. You need to be outside. You need to have like an actual burrow, like cage, more or less. There's also no data suggesting they do it for climate, mostly just for desiccation. They don't want to dry out. So for that reason, there isn't necessarily a link, but yeah, we want to look to see if there is one. Maybe it just hasn't been discovered.
So they seem like are we good?
That's all the time we have for questions, but we'll have coffee in like 5 minutes, so feel free to ask questions then. So let's thank Dom. And I'll just wrap up these past 2 sessions with a little summary. I want you guys to think back all the way to almost like an hour and a half ago when we started talking about human disease and how important that is and how applicable that is to our everyday lives. We had Bradley who was talking about the application of his research, which is directly impacting the lives of those who are dependent on this antipsychotic medication, and how allowing people to have limited side effects while still being on this medication is really essential. And we talked about research in Haiti and how it's important to understand what's causing contamination in the water supplies, and that was really exciting to hear about how Grand Valley students can go out and really reach other people. And then we had Jonathan talk about his research in programming, which is really interesting, and talking about how we can only do so much in a lab, but a computer program that can help us really analyze our microarrays is really important. And then I want to shift gears to animal models, which are still really relevant and still really relevant to us and can be used as a really awesome tool. They allow us for a greater scope of impact. We can hardly see, sometimes when we're looking at a single organism, like, oh, we delete a gene in an E. coli, but it's really hard to see, but if you delete maybe that same gene in, let's say, like a cow, you might see a really big impact. And so that's really exciting to notice and see in our fruit flies and in our crayfish. And so Liz and Taylor both talked about how great fruit flies are as a tool to use to study behavior and genetics. And then Dom just talked to us about how great crayfish are and understanding the ecosystems that are in our own backyard and being able to predict some of the effects that these invasive species can have can probably help us maybe prepare for these upcoming things that might happen. So I'd like to close out with a big thank you to everybody, and now there is time for coffee and questions if you had any last questions. Thank you.